Welcome everyone to this month's Wrongful Convictions and Conviction Integrity monthly meeting. This month we are glad to welcome Dr. Daniele Podini from the Department of Molecular Biology and Biological Sciences, George Washington University, and also an ad hoc expert here with the Quattrone Center. We are so glad to have Dr. Podini with us. As always, we are proud to mention that this programming is conducted under cooperative agreement with BJA that funds our work to do this and programming like it. And as always, our statements, the views of our presenters, the statements they make are not reflective of BJA or their position. And I think that is all the introduction of front matter we need. So I would like to move it over to Dr. Podini to introduce the talk and tell us about DNA from fired cartridge casings and rootless hairs, technology and applications in post-conviction. Take it away, Dr. Podini.
Thank you. Thank you for the opportunity to the Quattrone Center. I really appreciate. Good afternoon, everyone. See if okay. Okay, I guess this is a disclaimer, right? I can go on to the next slide, right? Okay, good. So yeah, I'm going to be talking about DNA from cartridge casings and rootless hair. So let's start with some technical stuff. That technically the word should be cartridge cases, not casings, but everybody says casings, and so I can get confused with cases. So sometimes I'll say one, sometimes I'll say the other. Forgive me for this. And obviously we're going to talk also about rootless hair. So the learning objectives today are to explain modern forensic DNA technologies that can be used for low template DNA in degraded samples, which is what we're talking about when we're talking about the hair shaft or even hair and the cartridge cases. Discuss the recovery and the interpretation of DNA from these types of evidence. Talk about nuclear DNA versus mitochondrial DNA, and I believe this might be redundant for some of you, but maybe not for all. And then identify the best practices and limitations with the collection and testing and the interpretation of these types of evidence. And then talk about kind of how to apply a structured framework to evaluating post-conviction biological evidence that might be available, and to select an appropriate testing strategy for the case that you would be dealing with. So common questions for post-conviction testing are, does evidence still exist? Was it preserved appropriately? Is there an appropriate chain of custody? Was prior testing conductive? Was it conclusive? Were the tests that were performed scientifically sound at the time? And are they outdated? Is there something better nowadays? And was the entire sample consumed during the testing? And then if all this is true, if there is evidence and it was properly stored and there's a reliable chain of custody, would the results be relevant to the case that you're dealing with? And then also, can testing be conducted? Would you be consuming the whole evidence? And what are the chances of getting something useful? So let's start with some core concepts of DNA. So when I talk about the DNA to non-scientific audiences, I generally start by saying, imagine DNA as this enormous encyclopedia that is present in every single cell of your body. And then I say that each cell is going to open up chapters of this encyclopedia that are necessary to become the cell that it has to become. So a liver cell will open up the chapter on how to become a liver cell. A neuron cell will develop it into a neural cell after looking at those other chapters. But the important thing is that this encyclopedia, with a few exceptions, is present in its entirety in every single cell. And that's why we can obtain the same DNA profile irrespective of the cell type. And this is very different from fingerprints that obviously come only from fingers. And once I used this analogy with fourth graders, and I had a hand raised and said, "What's an encyclopedia?" So I feel that I don't know how long I will be able to use this analogy, even in court, because I feel maybe in 20 years people won't have an idea what it is. I'm confident that you still know what an encyclopedia is, but that's kind of the core concept. And obviously, nuclear DNA in general is very discriminatory. We generally, nuclear DNA can be differentiated in autosomal DNA, which is the DNA that's inherited by both parents, and Y chromosome DNA, which obviously is inherited only from father to son. There's also mitochondrial DNA. Here you see in this simulation, let me get the laser, these little organelles that should represent the mitochondria. And that's also useful in certain cases when the samples are very degraded, or human remains that are highly compromised, or old bones, or even hair shafts. Because there's a lot more mitochondria in the cell, and we'll talk a little more in depth about them later on, there's a greater chance of recovering that type of DNA for a series of reasons that, again, I'll talk about later when I talk about hair. Another concept that's important, trace DNA. Some people call it touch DNA, which is it's kind of not an ideal term, but in general, that refers to low-level DNA that can be often deposited by touching an object. So that's why, but it doesn't necessarily, it doesn't come from the touch necessarily. It can come because, it can come from sweat. It can come from somebody coughing in their hand, or picking their nose and touching an object, or it can also be transferred. And also mixtures. And the sensitivity of the methodologies today is so high that often you pick up DNA that's in very low amounts, and so you observe mixtures. And so that's something that's important when we think about old cases, and potentially DNA evidence from a previous case that wasn't completely interpreted because there was a complex mixture. So we'll talk about that too. So state of the science and technology overview. So the typical workflow is that DNA evidence is well, I should say, evidence is collected, it goes to the lab, it's screened for biological evidence. So anything that could yield a DNA profile is identified, is characterized. There's some presumptive and confirmatory tests that can be done on the evidence to determine the type of body fluid. Once the lab determines that this is potential evidence, is suitable for DNA analysis, then they extract the DNA, they quantify the DNA, and this is an important process because it tells us how much DNA there is. The new methods that weren't available even 15 years ago can enable us to determine whether there could be potentially a mixture between male and female. If the DNA is degraded, there's also a way to look at DNA degradation with these new quantification technologies. So that informs the next steps in the process. And then you do PCR. Now, everybody has heard PCR because of COVID, so I won't focus on that, other than it enabled us to make millions of copies of specific targets to make them easy to analyze through capillary electrophoresis. And then there's the interpretation, statistical evaluation, and obviously then it's reported. So the typical region that we look at for identifying an individual are called short tandem repeats, or STRs. And I'm confident that many of you are familiar with that. And what these are short sequences, predominantly of 4 nucleotides, so 4 bases, which are the components of DNA, repeated a certain amount of time. So in this example, we have 10 repeats. And the polymorphism, so what changes between individuals, is the number of times that this sequence is repeated. So in the example below, we have 11 repeats and 12 repeats here. So this could be, for example, the chromosome that was inherited from the mother, and the one down here, the chromosome that was inherited for the father. So this particular individual has one allele, which is, so the allele, the concept of allele is the way that region of DNA that we're targeting is. So another analogy. Let's imagine that there's a DNA region, a location, which we call also locus, that is specific for eye color. So the locus is eye color locus. And the allele is blue eye, green eye, red eye, assuming there was one, black eye, et cetera, et cetera. So the locus is the region. The allele is that the way that region is. So at this particular locus, the alleles are 11 and 12 in this individual. So what we do to analyze this region is we design, well, these are commercially available kits. Now there are primers, so short DNA sequences that are specific, that specifically identify this region on the chromosome. And with that, we can attach these and then extend and copy this region. So make millions of copies of this region and end up with these two fragments. The primer, like I showed at the beginning, has a little fluorescent tag attached to it that makes that detectable. And then after PCR, we run this through what's called capillary electrophoresis. And so this is a totally AI generated machine. They don't look like this, but because of rules, I couldn't use the true picture of how it looks. But if you Google ABI 3500, you can find how the true DNA sequencers look. And so what these generate is a profile. And this as an example, a completely made up example of the way a DNA profile looks. So the x-axis represents the size of these DNA fragments, and electrophoresis is a process by which we separate these DNA regions based on their size. And the peak height is a consequence of how much of that target was present in the sample when you set up the PCR. So in general, the typical STR profile contains a sex marker. So there's the X and Y chromosome obviously are very different. The X is way bigger, the Y chromosome is very small. But they have a region that actually is very similar. And there's a gene called amelogenin, which is responsible for our dentin in our teeth. And that's on the X and on the Y chromosome. And a region in this gene that doesn't code for anything, that's called intron. So that's not translated into a protein, but it doesn't code for anything, but it's in there. That has an insertion. So the Y chromosome has a little fragment inserted there. So when we amplify that region, if there's a Y chromosome, it generates a fragment that's slightly bigger, and so they separate. Whereas if it's a female, both X chromosome yield the same size fragment, so you would see only one peak here. And that's how you differentiate the sex of the individual. So here you see some different peaks, and these represents alleles of a certain locus. So for example, at this locus D3S11358, this individual inherited from one parent allele 15 and 17 from the other parent. If both, and this individual would be defined as heterozygote at this locus, whereas at D5, the individual inherited 11 from both parents. So both parents gave the same allele. And so that's why we have a single peak. And the individual would be defined as homozygote for this locus. So these can be then translated into simple numbers that then can be used to be uploaded into a DNA database. And that's kind of the advantage of DNA, that really it's an Excel sheet in the database, right? Obviously much bigger because there's millions of profiles, but it's really simple to store the information because it's essentially a series of numbers. And then you can imagine it as a barcode that can be then compared to a database, and you can identify, you can determine whether that profile has already been detected and uploaded into the database. And you can also determine the frequency of a profile. So each one of these alleles has a certain frequency in the population. So what you can do using the Mendel laws of inheritance, essentially, you can calculate the frequency of the profile. And so that's why you see what's called a random match probability. So you obtain a profile from the evidence, you compare it to a suspect or to the database, and you identify a person that can't be excluded from being a contributor to that profile. And then the scientist reports the probability of obtaining that same, of randomly finding in the population another individual that also could not be excluded from being a contributor to that profile. And so you see, 1 in a quadrillion, these numbers that are a full profile that are, also kind of unimaginable. If you think on the estimates are that there's been in the past 50,000 years about 110, 115 billion Homo sapiens walking across the planet. So when you get a number that's a quadrillion or quintillion, I don't even know if there's ever going to be that many Homo sapiens on the planet. If we keep going like this, probably not. Okay, I shouldn't say stuff like that. When we talk about low template DNA, so that's a full profile, what I showed you, right? Let me go back here. So you see all these peaks are nice and balanced. There's a peak at there's 1 or 2 peaks at every locus. So this is a nice clean profile. But when we're dealing with low amounts of DNA, that doesn't really happen. So we obtain partial profiles. There's dropout, meaning some regions don't amplify. There are things called like stochastic effects, which are kind of imbalance between alleles that come from the same individual. And kind of conceptually, imagine you have a bucket full of marbles, and you know that in this bucket there's the same amount of blue and red marbles. If you pull out 1,000 of them, you're gonna get close to 50-50. Maybe it's not going to be 500 and 500, but you might have 400 and 600 or 450 and 550. But if you pull out only 5, you might get 1 and 4, or you might get even 5 of only one color. And so that's kind of the concept of this stochastic variation. So the lower the amount of DNA, the greater are the chances that you're not able to get a good representation of both alleles or different contributors that are in that mixture. Like I said, partial profiles that yield a lower random match probability. And also sometimes you do get a profile, and now because the sensitivity is so high, the issue is not really anymore that whose DNA that is, but really how did it get there? So we've seen that transfer is possible, between individuals. If you have an intimate encounter, you might have some of that person's DNA on your hands, and then you touch an object, you may transfer that DNA onto that object, but that person didn't necessarily touch that object, and there's controversy on it, and there's a lot of studies on it, and it's not the objective of today's talk, but that's something certainly that may be relevant in some of the cases that you might be working on. So modern DNA technologies, well, what has changed in the past 20 years? Well, first of all, the number of loci that is generally analyzed went from 13, which was 13 or 15, which were the kits that were used, about 15, 20 years ago, to now 20, more than 20 loci. And CODIS, the database, is actually expanded from 13 to 20. So right now, the kits that are available have between 22 and 24 STRs, different markers that are amplified at the same time. So that increased the power of discrimination. It increased the power of the probability of deconvoluting a mixture. And so that really has expanded our capacity. And the kits are a lot more sensitive, so you can obtain a full profile with less and less amounts of DNA over the years. Another thing that has changed is YSTR analysis. So 20 years ago, it was just starting, and now it's kind of routinely available. And there's like modern kits that also have what are called rapidly mutating YSDRs, which have a good chance of discriminating between closely related male individuals. So, like I said before, the Y chromosome gets inherited from father to son. And so brothers from the same father have the same DNA profile, other than if there's a mutation. Mutations occur, but they're not that frequent. And so the scientists have identified Y chromosome that have a high chance of mutation, high chance compared to regular SDRs. Not that they mutate super frequently, but in 1 in 500 meiosis, they mutate something like that. And so by analyzing multiple of these rapidly mutating YSDRs, there's a chance that you may discriminate even between brothers. So that has a lot of value. And then there's probabilistic genotyping, and that is a game changer. And I'll talk about that in a little bit. And mitochondrial DNA sequencing has expanded. So now we can sequence the entire genome, and certain laboratories are available or are able to do that. And we usually focused on a region that was where we would get the most bang for the buck. The origin of replication has regions that are called hypervariable. And so by analyzing those, you get a greater chance of discriminating between individuals. But now we can sequence the entire genome. And although the rest of the genome, the mitochondrial DNA genome, doesn't have as much kind of variability per nucleotide, so per distance, but overall it has just as much variability, if not more, of the hypervariable regions. And so it has value, and by sequencing it, we can increase our chances of discriminating individuals. And then there's the single nucleotide polymorphism. So testing through next generation sequencing, instead of looking at SDR, so the fragments discriminate the individual based on the fragment size of these regions, you can look at single changes. So an A that switches to a T, or a C that switches to a G, spread all across the genome. And that's kind of the scientific basis for forensic and investigative genetic genealogy. That there's a big hype on it, and obviously it has value. So probabilistic genotyping. So back in the day, you could really only interpret mixtures that had maybe two contributors, or maybe they had more than two contributors, but there was a clear major contributor or a clear minor contributor. So you could get information out manually, let's say manually, for limited complexity mixtures. But probabilistic genotyping has really changed that. It uses statistical modeling and certain algorithms that are obviously based on chemistry and amount of DNA, and on genetics and on allele frequency, to obtain what's called a likelihood ratio. So the concept has changed. So it's not anymore you obtain a profile, or even a partial profile, or you can deconvolute a mixture, and you'll say, okay, this person can't be excluded, and you provide a random match probability. So the probability of finding a random person that also can't be excluded from being a contributor to this mixture is 1 in whatever. Now the concept is different. Now we're comparing the probability of observing that mixture given two different hypotheses. One is that the person of interest is a contributor, and the other one is that the person of interest is not a contributor, and the contributor is somebody else in the population. And so it kind of shifted, and it's essentially Bayesian statistics. And so you provide a likelihood. So it's 100,000 times more likely to observe this mixture if it's a mixture between the suspect and the victim versus being a mixture between an unknown person and the victim. Or you could say, if there's no known contributor, maybe it's a 4-person mixture, and you can say it is 1,000 times more likely to observe this mixture if it's a mixture between the person of interest and three unknown contributor versus it being a mixture of four unknown contributor. And that's what you provide as statistical information to the jury, essentially. So now let's dive into cartridge case testing. Okay, so historically, these were examined primarily through, for firearm and tool mark methods, so to determine whether they were shot from the same gun. But modern testing has shown that DNA recovery is possible. And so we can identify the person that touched the bullet as it was being loaded, most likely. We don't know, obviously, who shot the gun, but we can say that DNA is present on that case. It may be important in certain cases. It may be irrelevant. And also in cases with like a drive-by shooting where there's nothing else, that can provide information that can be useful to the investigation. And so that's why it's relevant. But so how is the DNA transfer? Well, when we load a gun, we touch the bullet. There's a lot of pressure that's put in with the fingers, and as you compress, there's a spring inside the magazine, and as you get deeper and deeper, it becomes harder, and so you put a lot of pressure. And so there may be DNA deposited on the magazine, as you see on the cases. And where does the DNA come from? It comes from sweat. Like I said earlier, if you cough on your hand, there might be some saliva, some mucus, and even some skin cells. Although the skin cells per se are highly keratinized, and the DNA in the skin cell is kind of like the hair shaft that is very degraded, but it still can be obtained and identified. What are some of the scientific challenges? Well, there's heat, obviously, that's generated, the high pressure. The metal surfaces are also very challenging, particularly copper. And I have a slide on that. In the next slide is going to be about that. The environment, the cartridge case may be lying on the ground, may be very humid, or it may be raining, there may be a lot of sun. So there's a lot of things that can degrade the DNA on the case. Mixtures are common, so even if you obtain a DNA, maybe a complex mixture that may be difficult to interpret. Probabilistic genotyping is helping a lot with that, but it doesn't solve every mixture, and some are very complex. Like I said, it may be low-level DNA, so it may be challenging. And so we don't know necessarily when the DNA was deposited. That said, if there's an alternative suspect in a case, it may be very useful, or if there's a hit on CODIS, that also may be very useful. The person in some way might have been related to that case, so that might be useful. So brass is kind of the most common alloy that is used to make cartridge cases, and brass is made of copper and zinc. And copper actually creates these reactive oxygen species that degrade the DNA. And this has been known for millennia. The Romans actually would put their water, store it in copper pots or bronze pots, and bronze is also an alloy. It's mainly copper and tin, and so by storing there, the copper actually, because of this property, it actually kills bacteria as well. There's also some studies that have shown that like in hospitals, by replacing the doorknobs, the bed frames with copper, pure copper, that reduced the amount of bacteria in the hospital. So it's just expensive, but it actually has, it's a known phenomenon. And so particularly if there's water on it, because these oxygen species, reactive oxygen species, form in those cases. So Here's an experiment that we did in our laboratory here. We had cartridge cases made of copper, aluminum, nickel, and steel. And we had them handled by individuals for a certain amount of time, and then we let them sit for a certain amount of time, and then we collected the DNA from these. And here are the results. So as we progressed with nickel, aluminum, and steel, progressively over time, yes, the amount of DNA that we recovered was um, reduced, but it was still a lot more than what we recovered from even just 30 minutes on the brass cartridge case. So this kind of was an initial study that we did in our laboratory and in collaboration with ATF that kind of triggered some of the studies that they did later to optimize a method to extract DNA from cartridge cases, and I'll talk about it in a minute. So kind of this was the initial study we did a long time ago, and then kind of ATF, particularly Todd Bill, did is optimizing a method to extract DNA. But the first step is really the collection. So we need to talk about that because it may be relevant to the case. So it's important that the evidence is collected as soon as possible after the shooting, that is appropriately collected, meaning you have to wear gloves, you have to minimize touching the edges of the case. And ATF has developed a special collection device that allows them to store it. So the cartridge case is placed on this kind of plastic holder that has a kind of a post here where the cartridge case goes in, and then there's a cover that's put on it. And so even though this can move a little bit, it will never touch the inside of the cover. And so the side, and also there's a little piston here that kind of blocks the case at the center. And so this is kind of an ideal way to collect it, but if not, at least collect them individually. They should be placed in paper, so not in plastic, so especially if it's humid, so the DNA doesn't get degraded. Handle them minimally, and so all this has, are things that are currently being taught to crime scene investigator as they collect the evidence to minimize the chances of contamination or of destroying the potential evidence that is present on these casings. Then special extraction methods have been optimized for recovering DNA. One, we're using solutions that minimize the activation of these copper-induced reactive oxygen species that break down the DNA. So using a solution that is poured multiple times over the casing. And then there's a double swab that's being performed on it. And so this method was optimized. It's currently used by ATF, even though ATF continues to optimize methods. It's been published, and other laboratories, private laboratories, have optimized methods kind of using this as their starting point. And this is an example of the data, the increased yield that's been obtained from these with these new technologies. So this that you see in orange and in gray are the most recent methods that have been optimized, whereas this one, which is kind of the double swab, wet and dry swab with water, which has been kind of the classical way to, swab any type of surface. You swab it with a moistened swab, and then you kind of dry it up, and then you extract from those two. So if you're, any type of object from which you want to extract surface DNA, like a mouse, for example, you could use water, and that would be just fine. But because of the fact that we're dealing with copper here, you want to use this other process that has been shown to be a lot more effective. And so this is data from the publication from ATF. And these are data from a presentation from ATF in 2022. But we, I heard, so there's no other publication recently, but the numbers are very similar. So at the time, they had processed They had triaged 1,400 FCCs, fired cartridge casings, and tested about 800. And in 28% of samples, this yielded profiles that were suitable for comparison. 11% were profiles that could be uploaded onto NDIS, which is essentially managed what the database that is managed that CODIS accesses, so the National DNA Index System. Because from drive-by shooting, there's not always only one case that's found, one casing that's been collected, but there's multiple. The overall yield at the time was about 50% success rate, and now because the methods have been improved, I think it's a little higher based on direct communication with people from ATF, but I haven't seen a publication on that. So this at the time resulted in 40 confirmed CODIS hits since 2019, and that number is much higher right now. So much that the ATF has invested a lot of money. They're opening up a new facility where I actually have former students that are actually setting up this new lab that's going to be automated, and it's going to be centralized. So I think it's in Wichita that they're going to be receiving cartridge casing from all over the country, and there's going to be a high throughput lab, all because of this new method that's been optimized that is so efficient in closing case, well, in investigations. Now, here is an example of, and this is a completely simulated example of what you would see for a partial profile. So you have higher peaks for the smaller fragments because the DNA is very degraded. So as you're looking at bigger and bigger fragments, the chances of finding an intact one are less and less. And so that's why you see a decrease in the height of the peaks, which, like I said earlier, is a consequence. It's a kind of a proxy of how much DNA of that particular size, of that particular allele, is present in the sample. And to the point that sometimes you get dropout, partial or complete dropouts, meaning you don't see anything from the you don't see anything at that locus. So you obtain partial profiles, you see dropout, sometimes you see mixtures, so you see alleles coming from a third contributor, like in this case, or second or third contributor. So there may be some issues like how did that DNA get there? Was it through direct contact? Was it through indirect contact? Or some call it primary or secondary transfer. I prefer say direct or indirect. We don't know if it's indirect, if it's secondary and tertiary, we don't know that. So I just prefer saying direct versus indirect. And so these are kind of challenges in the interpretation. So some of the things that we have to consider when dealing with cartridge cases is were they collected appropriately? Were they preserved appropriately? What kind of testing has been done? And if testing has been done for conventional firearm investigation, so looking at, for example, the firing pin or the extraction marks that are during the firing of the bullet, of the round, these markings can help us identify the gun. So if these types of tests were done, how are they done? Were the analysts wearing gloves? Were they thoroughly cleaning the case before loading it onto the microscope? So these are all important questions to know in order to determine whether that case, the analysis of that case is useful. So we should see if we can find SOPs. What is there? Obviously the chain of custody has to be maintained appropriately. Where were they stored? In what containers? What temperatures? How long ago were they available? How much time how long have they been stored? So how old is the case? And how much time passed from when the shooting occurred and the samples were collected? This is also important. And then other questions, is there a known suspect? Are comparisons available? Of course, if you yield a profile that's uploadable to CODIS, then that in itself is useful, but you don't know that going in. So that's important. And as you saw, it doesn't happen often, right? There's a chance, but it doesn't always happen that we get a profile that's good enough to be uploaded into CODIS. Okay, so that's all I had on cartridge cases. Should we do questions now or do everything at the end?
That's a great idea. Let's take a pause for questions. Folks can put them in the chat, and we'll also take time for questions at the end. So if there's something that you want to ask, you can go ahead and put it in now. And we'll give folks about 20 more seconds to do that.
All right, we can also, you can type them in and then we'll answer them at the end. I just wanted a break to drink some water. That was a good excuse.
It was. All right. So we'll continue on with hair shaft testing. As you have
I'll continue.
questions, please go ahead and put them into the chat.
All right, so let's shift and now talk about testing from hair shaft. And so in my lab right now, I actually have I just completed a project optimizing hair extraction that was funded by the FBI, and I'm starting a new one, still funded by the FBI, on trying to see if I optimize an extraction method, and now we're trying to see if we can repair the DNA from the hair. It's a tough project. The chances of it working are low, but it's actually what I'm doing now. And then I have another grant from an IJ that's still ongoing, that we're looking at extracting DNA from the hair, but not looking at SDRs, but rather SNPs. And so I've actually been working on this quite a lot and had some interesting data. But today we'll focus primarily on the SDRs part, but I'll give you kind of a general overview as well. So hair evidence can appear frequently. It was used a lot in the past, and hair comparison was used and overstated at times, and there's been some mistakes, and so the value of it has progressively declined. If there's a hair root, then we have a good chance of getting nuclear DNA out of it, but kind of historically, if there was just the shaft, what was useful really, what was possible really was only typing mitochondrial DNA. Nowadays, with modern methods, we are seeing that there is nuclear DNA in the shaft. It's just very degraded. So you have to optimize a method of extraction that captures as much degraded DNA as possible, and then target loci that have small alleles. So basically, the primers that I showed you earlier try to design these primers that are as close as possible to the repeat unit so that you amplify fragments that are small, and then you have a greater chance of being able to detect them in the DNA extracted from the hair shaft. So again, best practices, the hair should be collected either wearing gloves or maybe with forceps that have been cleaned. They have to be packaged individually when possible, using paper rather than plastic envelopes, or plastic bags like Ziploc bags are not good, especially if there's moisture. And then separate questions from knowns, and obviously everything has to be documented well, the location and who collected it, etc. So these conditions have to be met in order for the hair to be useful down the road. So the hair shaft. So what happens is here we have the cells at the bottom of the bulb that are kind of stems, in a way, stem cells, meaning that they're not super specialized, although they're specialized enough to just be useful there, but they continuously divide and they kind of push the stem up. And as they become part of the stem, they produce keratin, and this keratin breaks down the nucleus and kills the cells and kind of degrades, the nuclear DNA. But in every cell, there's one nucleus, but there's hundreds of mitochondria. And mitochondria, I told you, I promised you I was going to talk to you more about them, are these little organelles that are responsible for producing energy. So they have a important value for our organism, for for eukaryotic cells in general. And but they have a bacterial origin. So I don't know, a billion years ago or something, at a certain point, there was a cell that phagocytated, that's the word, but englobed another cell, a bacteria, and instead of killing it, they decided to be friends and to form this symbiotic relationship. Eventually, these bacteria became mitochondria, but they kept their DNA, which is important for its function. Obviously, it needs DNA from the nucleus because some of its functions are dependent on that. And obviously, it has, it wouldn't be able to survive alone outside of the nucleus. But some of those features are some of the functioning properties of the mitochondria depend on this mitochondrial DNA genome, which is a circular genome. And that's why we know that they have a bacterial origin, because bacterial genomes are circular. And in any mitochondria, there can be 2 or 3 of these circular genomes. So it's a question of numbers, meaning, we could have 1 nuclear DNA and 1,000 mitochondrial DNA. And it's also circular, which makes it a little more resistant to degradation. So these 2 conditions make it so that there's much higher chance of getting mitochondrial DNA as opposed to nuclear DNA when a sample is very degraded, or from a hair shaft. As the hair elongates, grows, that's the easy word, grows, it's exposed to sunlight, UV light, that breaks down the DNA. It's washed, there's hair treatments, and all these things break down and degrade the nuclear DNA in the hair shaft, making it very difficult to recover. When you analyze the DNA, also there may be some contamination on the surface if it was collected from outside, if it was touched, if it was ripped. So what you want to do is, obviously, you have to decontaminate it before you extract the DNA. So there's procedures to do that, and I'll mention them as we go on. This kind of I'm just going to skip this one. You'll have it, and I'll just talk about these things. And this slide is a lot prettier. So. Historically, when we were focusing only on mitochondrial DNA, mitochondria are inherited from mother to offspring. So mitochondria are present in the egg cell, and when the sperm fertilizes the egg cell, the nucleus of the sperm cell fuses with the nucleus of the egg cells, and the mitochondria that are present in the sperm, and they're important because they provide the energy for the sperm tail to move and kind of to propel the sperm for its mission, they get all broken down. So we inherit the mitochondrial DNA of our mother. So it's somewhat specular to the Y chromosome, meaning that it's inherited from mother to offsprings, but both men and women have mitochondrial DNA, so it's useful in that sense. We're not looking here at STRs, but we're looking at SNPs. So these single nucleotide polymorphisms, so these changes in the sequence. And these are a consequence of the mutation rate. So the mitochondria, like I said earlier, are bacteria originally. And so when they replicate, their DNA replication mechanism is very similar to that of a bacteria, which has some less efficient repairing mechanism. So our own DNA in our nucleus, as cells duplicate the DNA, preparing before dividing, they have ways to fix it as they copy the DNA. There's some errors that occur, and then there's ways to repair it. Obviously, it doesn't work perfectly. That's why we can evolve. Evolution is there because of that. But they are a lot more efficient than bacteria. And so what happens is that the mutation rate within the mitochondrial DNA is a little bit higher than with the nuclear DNA, especially in those 2 hypovariable regions that don't code for anything, and so mutations there don't affect the survival of the mitochondria. So there's a lot of mutations that occur there, and they can be used to compare two different individuals. Of course, if they're maternally related, they're going to have mostly the same profile. There's also a phenomenon that complicates interpretation that's called heteroplasmy. I won't get into it unless you have questions after it about it, because somebody maybe have encountered it. But anyway, what we've been focusing on is trying to instead get nuclear DNA and look at STRs or SNPs from the nuclear DNA, because they are a lot more informative than the mitochondrial DNA. So the process out here, going back to the process here, we document before collecting, and that's true for any type of evidence. They have to be thoroughly cleaned. So what we do is we treat it with certain detergents, a very mild detergent, and also some enzymatic detergents that clean up the DNA. Then we extract the DNA, trying to maximize the recovery of a small degraded DNA, and then we either amplify for SDRs, or you can actually amplify it for mitochondrial DNA and sequence those. And then obviously, if you obtain results, you can compare it to references or a database if you're looking at SDRs. So right now, the success that we've obtained is obtaining a profile from these hairs, and that's true also for laboratories that I've seen that have validated procedures for this, is around 20 to 35%. The chances of getting a partial profile is very hard to get a full profile, so like I said, some laboratories, and we can talk about it later, have validated protocols specifically for hair shaft. And this is some data that from our lab, from the FBI grant that I told you, we tested 130 samples. And here you can kind of see the breakdown. This is with one kit from a company that's specifically designed to look at very small DNA fragments. This is instead with a more modern kit that has fragments that are very big, and so we almost never get anything from these fragments, but we have a certain amount that are designed to be very small, and so we're getting some yields. And here you see the yield. So in about with this method, about 21% of the time of these sample, we got 30 or more alleles, and 20 to 30 alleles about 7% of the time. Most of the time we got this, and sometimes we got nothing. So that's kind of what we're getting from the method that we're testing, just to give you an idea. And here are some examples of profile, and these are true profiles from our lab. So the ones I was showing you earlier were simulated, but as you can see, as this is with Minifighter, the first kit that I told you that has fragments that are designed specifically to be small, you see how this particular locus is amplified very efficiently, but this is very, very low. And this is because these are smaller than these, and the DNA is very degraded. And you see this pattern in these different hairs. And so this was a hair that yielded a lot of DNA, but this was one that instead yielded a lot less. But what's called the degradation index tells us that we're still able. This is information that we're getting from the quantification. It tells us how degraded the DNA is lower compared to this, so we're getting more information. This is an example of a complete dropout of this particular locus. Here is another hair with the PowerPlex kit, which is the second one I showed you that was on the right of the first slide. And as you see, we're seeing results from loci that are small, but it's a complete dropout for the larger loci. Instead, this one had a slightly lower degradation index, and we're getting a lot more DNA from this sample. So this would be something that we would be probably able to upload on CODIS. So it's a good profile. But like I said, it's not the norm. In our study, excuse me, in our studies, we've seen that there's a lot of variability in the individuals, and it's very hard to predict. We try to look at morphological features that can associate the color of the hair or the structure of the hair or the ethnicity of the individual with the chances of getting a profile that's useful, but we, at least with the limited amount of samples that we had that actually yielded a profile, we weren't able to find any kind of metric that can help us predict the chances other than a slightly higher chance, it looks like, of getting something if there's no medulla in the hair. So the hair has a medulla, and well, humans sometimes have, sometimes don't have a medulla. Sometimes is a partial medulla, whereas most animals have a medulla, which is kind of a inside hole inside the hair. If they don't have it, there's a slightly greater chance, but we need to test more samples to determine whether it's statistically significant. And this is really then what's important really is that profile. How rare is it in the population? And so these are all the hairs that yielded a profile, and as you can see, some of these, 1 and 2, because we got only an allele or a very partial profile. So the frequency of the profile from this particular donor was, we yielded 3 alleles, and it's 1 in 372 individuals. But some of these hair, this particular one, for example, yielded 15 alleles, and we got a random match probability of 1 in 10 million. And so that could be useful in a case, for sure useful for exclusion at the least. So if you find yourself in a situation like this particular hair might be useful. If you have an alternative suspect that matches this profile, then it's even more helpful, not simply an exclusion. The best one was this donor that had, a profile that had some rare alleles, and it yielded a profile that's 1 in 205 billions. So that is kind of, I'm not saying enough to identify an individual, because you can't say that, but it's certainly be very useful in a case. Okay, so now let's talk about next generation sequencing, and this is what we're doing with the NAJ project. So what we're looking here, we're using a method that's called capture hybridization. So we have some RNA probes that we mix with our sample once we extract it from the hair shaft, that go and kind of bind to specific targets throughout the genome. And these have little molecules that we can use to attach magnetic beads and then pull these out of the solution and then sequence them. It's not something that you can right now use in a crime lab for routine cases. It's kind of cutting edge stuff that still needs to be optimized, but it's yielding some interesting results. And an alternative method that's being used, and it's actually been used in some cases, is to do the whole genome process. And the reason why we're doing the former that I've described is that we're trying to do something that can be more manageable cost-wise, and that can be implemented more routinely in crime labs. And so that's kind of why we got the funding. But I think as the cost of sequencing has been going down and down really, really steeply, it could be that going forward, just the whole genome sequencing might just be the way to go. So what we're looking here is these single nucleotide polymorphisms. So these little single base changes throughout the genome that can be used for identifying an individual if you have a direct reference. But you could also use it for forensic genetic genealogy. So you can you do the whole genome, or you target these specific regions that can be used for familial search. Oh, sorry, that's not the right term. It's basically the same SNPs that are used by 23andMe or Ancestry. 23andMe, yeah, they went bankrupt. But other companies like Ancestry.com and other vendors of that generate these SNP profiles, and some of these can be useful to look at the ancestry of the individuals, and you can also look at the if you type the same SNPs across individuals, you can generate a likelihood of them being related or not. And so people have been, doing these tests. They're sending their DNA into 23andMe or Ancestry.com, getting that data, and then uploading it on GenMatch, which is kind of a free server where you can upload all your SNPs or your profile, and then see if you have any relatives, second degree cousin that you didn't know about. And there's a lot of controversy there. There's a lot of issues with people discovered that they were half siblings with their brother, or also that maybe their father was a sperm donor, because they have a lot of siblings out there in the population. So there's a lot of consequences with that, but it also has enabled closing cold cases like the Golden Gate, the Golden State Killer, and others. So it can be very useful for that. And this is an example of a case that where whole genome sequencing from hair shaft was conducted, and it was admitted in court. There was a lot of debate on it, but they used whole genome sequencing. So that's an approach too, but it's very expensive, and again, you need a reference really if you want to be doing the testing. So considerations on hair, okay, for post-conviction testing. First of all, we want to look at how relevant the hair that you have available can be to the crime. If it was found in the car and the car has, is not relevant to the case, maybe it's not useful to test. But if it was found at the crime scene on the body of the victim, it may be you, and there's been an altercation, it may be in the hands of the victim, then it may be useful. Would an exclusion undermine the original theory of guilt? So that's something that you should consider, and maybe use that as a way to advocate in favor of testing that particular sample. Is there a suspect? Because if you yield a profile that is not suitable for uploading in CODIS, it may not be useful if you don't have a suspect to compare it to. Or if you do have it and you upload on CODIS and there's no hits, then what can you do with it, right? If you have a suspect, an alternative suspect, you can use it to compare it, and that might be relevant to your case. Was testing done? Was it used? Microscopic hair comparison. Was it done? Was it used in a trial? Were the conclusion overstated? So is retesting it relevant because the person was convicted based on those tests? Is there a root? If there's a root, if it was ripped, then there's a much greater chance to getting an STR profile. And has Mito been tested previously? Are there, I guess this is redundant, right? Are there reference available for the victim, the defendant, but also other people that may lived with the suspect, with the victim at the time, and so maybe their hair was there because of that, and so that might be useful to eliminate the individual as a contributor. So these are all things that should be factored in when you think about testing. Another important thing is, if you have to select the laboratory, you want to make sure that the laboratory that is fit for purpose, right? Do they have protocols that are developed specifically for that purpose, whether it's cartridge cases or hair shaft? And because the nature of these samples, you need specific protocols that maximize the chances of obtaining a result. And so do they have these protocols? Is the laboratory accredited? And you should be aware of the limitations. Like I said, it doesn't always work, and the probability is probably there's probability that it won't work is slightly higher than it will work. And then, if the casings were treated, were cleaned, were touched with no gloves, or if the hair was badly preserved, you might not get results. And so you have to be aware of these limitations before going into it. And then also that the laboratory has CODIS access. This will simplify your things. If the laboratory has CODIS access, a private laboratory generally can partner with a state lab that has CODIS access, and that way you can upload your sample onto the database for comparison. So overall, this is a slide kind of summarizing a little bit of what we've talked about. So first of all, for post-conviction testing, you have a case. You want to see what evidence was collected from that case and what was tested, and what was testified in court about, what was relevant, what was not relevant. And once you have that in mind, if you think that there's potential evidence that could be retested, then I would recommend talk to somebody that has experience with a particular test you want to do, so that they can tell you whether there's a good chance of getting something, whether it's relevant, and go in with as much information as possible. So when was it collected? How was it stored? Was it tested? Do you have that data available so that the expert can see it? Then with them, identify probative evidence, develop a testing plan, and identify a laboratory that can do that testing, knowing what you expect as you're going, to have a plan of what you're going to do with what you get, based on what you think and hope you're going to get. And so that's kind of a slide kind of summarizing all that. And with that, I'm done, and I'm happy to answer questions. So
Thank
should
you
I
so
stop
much.
sharing? Or, well, I'll keep the slides up for now in case somebody has a question that we want to go to. Here's my email, and obviously you can go through the Quattrone Center if there's something specifically that you need through them. And I'll let Adina talk about that part.
Thank you so much, Daniele. So, we have some questions in the chat that I'm going to ask aloud to you in the time remaining. We will have, at the end of our time together at 3:15 Eastern, just a minute or two before, a survey that will deploy. So please keep a lookout for that. And I know a lot of folks are seeing fired cartridge casing or case issues come up in their work. If you have specific case questions, email me. We'll get you set up with some advice. So, to Dr. Podini, just going down the list of questions here. First, can you talk about testing bullets themselves instead of casings? And we'll differentiate between bullets that have hit a body and bullets that have not hit a body.
Yeah, so if they hit a body and then they went out of the body, if they hit a body and you collect it from the body, you're going to get the DNA of that person. That's it. I mean, there's so much of that DNA that even if they're assuming there's any traces of the person that touched it, it's going to be overwhelmed by the DNA of the person. The heat, the moving inside the barrel that's generated, the object that it hits, the chances of getting anything off of the bullet are very, very, very low. I don't know personally of any research right now. I can't think of any research that's been done off of the bullet. So I can't really, quote a study that's been done on that. But if I had to bet, the chances are very, very low.
And as a follow-up question to that, what about unfired bullets, such that maybe somebody might have dropped or emptied a magazine at a scene? Can you speak to testing full bullet and casing together, not run through a gun?
Yeah, so that's instead it's kind of on the other side. That's even better than the fired case, okay? Because that's not been exposed to heat. It's probably still in the magazine, or maybe it popped out or something like that. But that has kind of the same value, if not better, than the casing, the fired case, right? Because it hasn't been exposed to that heat, It has and the compression, because when the when the case kind of when the fire pit hits the kind of the igniter, the the gunpowder starts burning and expands it, there's a lot of pressure there and heat. So that doesn't help the DNA that's on the surface. So that process hasn't occurred. So you have the same chance. And some of the bullets itself may not be made of brass, may not be copper, may be only lead. And so that's even slightly better than the brass itself because there's no copper. So in that sense, you have a shot at it, definitely. And there's all the collection part and how it's been treated. That obviously stands. If it was treated and cleaned and all that, then and collected without gloves, then obviously it's not going to be that useful.
Thank you for that. Another question we have is specific to the ATF study where they reported a success rate. In reporting
Mhm.
or calculating that statistic, did they differentiate between the different composition materials of the casings they were testing?
So the specific that we were talking about, I am not 100% sure, but I think it was all brass because nickel and aluminum and stainless steel are not common at all. So the most common type of casing is brass. So I'm pretty sure it's only brass, but I can't remember. But there's a publication, so you can look it up, and there's probably something more recent. So these were the results of the study that they did on the cases. But the protocol that they're doing is from this paper here. So you see it's Todd Bill et al., an improved process. And I think they were all doing it on brass.
And
Because if it's something else, then your chances are higher.
if folks need access to academic papers, that's another resource that Quadrone Center can provide. So please do reach out if you're looking for resources. We're available to help you. Let's see, we have about 3 minutes left. You'll see that the webinar evaluation has just popped on your screen. If you'll please fill that out. And we've got some other Quadrone Center folks here on the call with us. Taylor and Marissa, our ad hoc DNA expert, Angeli Rana DeVay is on with us. Marissa or Taylor, I want to kick it to you to announce what's coming up in the next rounds of programming. On the DNA side, in September and October, we'll be presenting a two-part webinar on forensic investigative genetic genealogy, or FIG. We will be talking about both the biology side, conducting the SNP testing that Dr. Podini was explaining, and also talking about the genealogy side, or what happens once that profile gets uploaded into one of those genealogy databases.
Yes, we've also got a wonderful webinar by Matt Dietz next month. We're going to be talking about disabilities and mistreatment when police interrogations and interactions. So if you're interested in that, or you have a case with kind of details similar to that, please join us for that next month. We're also planning a half-day tech conference in November. So after those FIG webinars that Adina mentioned, we'll be doing a half-day tech conference that's organized by our data consultant, Chris. That's going to have a lot of vendors, a lot of things that you should be looking out for, things that you can implement in your day-to-day. So keep an eye out for those as well.
Thanks, Taylor. And as one more reminder before we go, the Quadrone Center is your training and technical assistance provider for DNA support services. So we do education and training just like this. We have a whole playlist of DNA related webinars that we hope that you'll watch and learn from. And we also offer consulting with Dr. Podini, with our in-house consultant, Angeli, with a whole suite of other folks that we have on demand to help you achieve your goals in your case. We're so glad you're here. Thank you for joining us. Have a wonderful rest of your day.